happy today to be joined by Tracy Feldman, who is an associate professor at St. Andrews University in Laurinburg, who's come to us today to talk about leaf miners and many other small insects and the often overlooked biodiversity here in North Carolina. And so with that, Tracy, I'll turn it over to you.
Okay, first I'd like to thank the environmental educators of North Carolina. I'd like to thank Lauren Pyle for having me here. And so before I talk about an unknown world of small creatures, and we'll talk about some of those small creatures for sure, I want to start with a quote by E.O. Wilson. "A lifetime can be spent in a Magellanic voyage around the trunk of a single tree." And the reason why I start with this quote for two reasons, two big reasons. The first one is that what it says to me is that we don't fully understand the biodiversity of Earth yet. There's still a lot we have to learn. And the second thing that's important about this quote is that in order to learn about most of the biodiversity of Earth, we have to look closely because most of the things on Earth, most of the life on Earth is small. And so looking closely is important. Can everyone hear me okay? If people want to ask questions, please feel free to use the chat. Someone already has used the chat at one point, but I guess we'll talk about that later. I don't know if I'll be much help with biocontrol, but I can talk about that later. And then also, if people want to interrupt me with questions, you can feel free to do that too. All right. Oh, wait, that's not. All right, so we'll click onto the actual thing. All right, so I'll talk about some overlooked biodiversity and ask some general questions of you guys. So why should we catalog biodiversity? And why here in particular, why it might be useful to catalog it here. And then I'll talk about lifestyles of the small and squishy. So these small and squishy organisms are leaf miners. In particular, I'll use them as an example, but there are many other types of insects that are very small and squishy. And I'll talk about what leaf miners are. All right, and then I'll talk about some discoveries so far. So new locations for some of these things, new host ranges, which means new food plants that we didn't know they ate before, and then new species, entirely new species. While we're talking about new species, I want to talk about how we name new species, like what's involved in the process of determining that a species is really new and how do we actually name it. And then what you guys can do to look for and find leaf mining insects and other small insects in your yards and wherever you have access to at this time, and how you might report that to certain platforms online to so that you can contribute to scientific studies, citizen science. So I'll start with a question. These are pictures of a small fraction of biodiversity on Earth. And I wondered why we should catalog biodiversity. What does it matter if we know what other species are around us? Anyone have ideas about that? You can use the chat. You can unmute yourself and speak whatever you wish, but what do you think? Why does it matter? Okay, I have understand life. It can be an indicator of the quality of land and water. Yes, sure. So I've got, all right, to track populations, understand ecology, our world, indicator species. We are all in this together. Aldo Leopold, keep all the parts. Yep, all that stuff. So, okay, so what I'm hearing is that some of these species may be really important to the ecosystem around us. And if they're not there, that could indicate problems with the ecosystem, with the system. And we may depend on these species more directly or less directly, either way. And well, it's also cool to know what's there. And so, and maybe, and there's also redundancy in the system, someone said, cool. So yes, so there may be certain species that if they're gone, other species will fill that role, but there's only so much of a buffer, and that could be a problem if that buffer disappears. Okay, and so when one species is removed, other species are affected by that. Certainly, it might matter to know what's there so that we know something about the health of our ecosystem and if something's missing or not. All right, so we'll talk more about that. One other reason to know about biodiversity is that we're losing it very quickly. If you look at the top curve, that's roughly human population growth. It's a really rough sketch of population growth, and then underneath it is the sort of extinctions of organisms. So what we're facing right now is the 6th mass extinction in Earth's history. And it's the only mass extinction that it's the fastest one, and it's the only one caused mostly by one species, us, humans. In May 2019, the Intergovernmental Science Policy Platform on Biodiversity and Ecosystem Services put out a report suggesting that over 1 million species are threatened with extinction right now. And there may be more, and certainly there are species we don't even know about. But of the species that are well studied that we know about, over 1 million of them are threatened. And so that's a big deal. So right now we're losing biodiversity at a rapid rate. And some people talk about the insect apocalypse, where we're losing insects. We've lost about 30% of the birds in North America. So we're losing a lot. And if we look at where most of the biodiversity on Earth is, we call them biodiversity hotspots because they contain either the largest numbers of species, larger number than other places, or a larger number of species that happen to be found nowhere else. So they're called endemics. Endemic species are found nowhere else. So all these green areas represent biodiversity hotspots. They're often on coasts. A lot of them are in tropics, but not all of them. And either way, there's only, what you might take away from this busy figure is that there's 5 to 30% left of all of these hotspots. There's not much left. We've been knocking out most of these hotspots. And as someone's pointed out, there's actually a hotspot that's missing here, and it's the southeastern coastal plain. There's 2 hotspots that are missing from this figure. It's a little bit older, but it's the best I can do for what I want to say here. But there's one hotspot that's missing here, 2 of them, and one of them is the southeastern coastal plain. And I'll talk more about that soon. So I want to ask one other question before we continue, and that's if we preserve habitats, like a lot of people would say, well, what if we maintain habitats the way that ecosystems are typically maintained? We burn them if they're typically burned, or we do other things if other things are typically there. We get the dominant species like these longleaf pines, and then it's going to be a healthy ecosystem because other things will come. What do you think about that? Do we still need to know what species are there, what other species are there? Or why might we want to know if we do want to know? What do people think about that? All right, yeah, okay, so maybe there are species that move through and we wouldn't necessarily know that. All right, so maybe if we preserve land, what if it's too small of a patch of land and some of these things aren't on there? We don't know unless we catalog what's on there. We don't know what's using that property. Yeah, so I'll give an umbrella question that just sort of leads into what I'm thinking about here. Yeah, so and threatened species, invaders. Yeah, so we want to know about those. All right, so are apparently intact systems really intact? Well, how do we know if they're intact or not? We have to know that they're supporting the life forms and the ecosystem processes that they're supposed to. And so how will we know if a species is missing or declining if we don't know what's there? How do we know if a species is becoming incredibly common? Maybe it's an invasive species, or maybe it's a non-invasive species typically that's becoming overabundant in this ecosystem because it's disturbed in some way. We might not know that until we actually catalog the organisms in it, what's happening. Or we might want to know, as climate change happens, we might expect that some organisms are going to emerge later or earlier, or have a longer or shorter season, and that is certainly going to be impacted by climate change and maybe by the habitat quality, and we won't know that unless we know what's there. So all of these are arguments for knowing what's actually out there, because we can't really monitor changes in it unless we know what's there to begin with. Okay, like I said, we're one of 36 biodiversity hotspots in the world, and this picture just gives one small example of that for plants, and there's a reason why I chose plants. So this map is bluer where there's more diversity. So where there's more biodiversity, you see that it's bluer. So you can see that the coastal plain is where, the southeastern coastal plain is where the most biodiversity is. And this area in general is home to most of the biodiversity in the United States, at least, in terms of endemics. If you look at endemic birds found nowhere else, mammals found nowhere else, amphibians in general, reptiles as well, fishes. If you look at all those things, trees, the Southeast is really where it's at, the southeastern coastal plain. And why plant family diversity is so important? Well, a lot of the insects that we're going to talk about are relative specialists. That means they feed on some plant species and not others, some plant families and not others. And so that means that the more plant families you have, the more potential insect biodiversity there is. I have no map for insect biodiversity, but I can infer a lot of insects eat plants, not all of them, but many of them do, and a lot of the ones that do are somewhat specialized. And so this might also indicate the amount of biodiversity of insects here as well. And we have potential to lose that biodiversity. We have the 12th largest growth rate here in North Carolina, about 1% yearly, and that amounts to more land conversion, because as more people come, there's going to be more land converted, and land conversion is one of the main threats to biodiversity. Not the only, that and climate change are the two biggest threats, and then poisons are another one. We'll talk about that too. Most of the biodiversity, if you look at just plants and animals, disregarding fungi, viruses, and bacteria, of course, but looking at just plants and animals, insects make up more than half of the biodiversity out there. So there are large numbers of insects out there, and so we'd expect biodiversity to be big. And in fact, we just don't know what's out there. There's a lot of insects we don't know about. And in fact, plants can help maintain that biodiversity, and specialization can help maintain biodiversity. So I want to illustrate that here with this point here with this rough illustration. I know my art is kind of poor, but here's a plant, and you can imagine there are a lot of things that eat that plant. So here are some leaf-feeding insects, and mostly insects, but other things too might eat leaves. Deer might eat leaves. So we have feeders on the surface. We have leaf miners that are underneath the surface. We'll talk about what they are more in a second. And then gall insects as well that are kind of interesting. They inject chemicals in the plant to make them grow these balls that the insects live inside. There are sap feeders that suck the sap out of the plant on leaves and stems. There are flower feeders, stem feeders, root feeders. Many of these, but not all of them, are insects. Then we've got pollinators, also mostly insects. Seed dispersers, fruit and seed feeders, some of which are insects. Protective ants that protects things like aphids and scale insects. We've got mycorrhizal fungi that associate with the roots, clearly not insects, but at the same time, some biodiversity maintained by this plant. We've got predators and parasites of all of these things. And some of these are specialists, some of them are not. We've got predators and parasites of all these. We've got predators and parasites of predators and parasites. So all of this can be maintained. So this plant has its own suite of associates and decomposers as well. Each plant has its suite of associates, and the next plant over of a different species might actually have some of its own suite of associates, and many of those may be different. And so we can see how biodiversity can sort of balloon once we've got a plant there, we've got a whole bunch of things that use it. All right, I'm going to focus on leaf miners as an example because I've been doing a lot of work on those for the past 6 years. And I'll show you some of the discoveries I've made with these. But before I do that, I want to review the insect life cycle, because that plays a role in what we're going to talk about. Many of you may be familiar with insect life cycles. There are three rather different kinds of life cycles that insects have more broadly. And I'm going to focus on one of them called complete metamorphosis. And that's because all the insects groups that are leaf miners have complete metamorphosis. So here are two examples of that. You start with eggs. So these are moths. They start with eggs. Flies start with eggs too. And then they hatch out of the eggs as larvae. And they might go through 3 stages shown here in this fly life cycle. Or in moths, you might have anywhere from 4 to 7 life stages in the caterpillar stage. And then they pupate. Now the pupa is where they change from the larva to the adult. And so there are a lot of big changes happening here. They're losing parts, they're gaining parts, they're changing around a lot. Their body's changing around. And because of that, they don't move very much. Because they don't move very much, they are vulnerable. And because of that, they often stay hidden when they're in the pupal stage. And there are some interesting adaptations to hide pupae. We'll talk about a few of those later. All right, and then they come out as an adult and the life cycle starts over again. So these are just 2 examples of 2 of the groups that are the main leaf miner groups, flies and moths. There are 2 others. So what are leaf miners? Leaf miners, imagine you holding a leaf in front of you and you turn it on its side so you're looking at the base, like as a thin strip. And that top surface of the leaf is here. And imagine that's like the ceiling or something. And then the bottom surface of the leaf is here, and that's the floor. And these leaf miners are mining their way through the inside. They're eating the tissue in between. So it's in their larval stages that they do that. The other thing is that it's a lifestyle. It's not one taxonomic group. So beetles, flies, moths, and wasps do it. But not all of those. Not all beetles, not all flies, not all moths, but certain groups of them do it. And because that area is so thin and so small, these leaf miners and gall insects too are often very small. They're often very small. So their sort of modus operandi is stay relatively small, stay relatively hidden. And often their life cycles are quick, but not always. So that's another thing. They can escape detection that way. Here's a leaf. This is a holly leaf, an American holly leaf, with 2 leaf mines on it. This is a mine made by a larval fly, Phytomyza ilicicola, and here's what the adult looks like. And then over here on the same leaf, you have a leaf mine made by a moth. And it's white because they put all their poop out the side, out a little hole in the side. They're actually practice sanitation. The fly just poops behind it as it goes, never look back. And so here's what the adult moth looks like of this one, Rhopoboda finitamina. Here's what it would look like in the wild. You'd come upon a leaf, and if you look closely at that leaf, you see, wow, there's 2 kinds of damage here. There's a leaf chewer that chews a hole through the entire leaf and breaks the surface. They either feed on they break one surface or another, or both. But then you have 2 miners as well, that the leaf surfaces are intact, but inside, that's where the feeding damage is. All right, so this is what you guys are going to look for too. You're going to look for leaf mines and other things too. All right, so I'm going to talk a little bit about specialization. Yes, there's a snail in here, and that's kind of cool, but the plant is what I'm interested in, and that's a winged elm. Winged elm is kind of a weedy plant around here. They're really common trees. They're kind of everywhere around here. And there are many leaf miners that specialize on them. There are other things that are there too. So basically so there are lots of herbivores on winged elm. And I'm going to just talk about the leaf mining insects, which are just a few of them, but there's still many specialists. And so it's kind of cool. This is just one group, and there's many specialists within this one group. This Agromyza aristata, mostly an elm specialist, also feeds on sugarberry as well. And then Stigmella apiciobella is a moth. So there's a fly, there's a moth. These make trails. These mines look like trails or linear mines. There are also blotch miners, miners that make like a blotch-like shape damage. This is called Cameraria ulmella. This also is recorded on oaks, but that may not be true. It might end up being a separate species that's on oaks. Hard to say for now. We hopefully will find that out. Brachus species, that's a beetle. That's another blotch miner. So there's another beetle that's a generalist that feeds on elm as well. So I didn't mention that here. There are these two that are called tent miners. The tent miners scrunch up the leaf surface on the top, and then on the bottom, they sort of have a wrinkled bottom surface of the leaf. So they look like little tents, and that's where they mine. There's another one, Ectoedemia ulmella, another moth. There's another moth, this Buculatrix species. Buculatrix are the ribbed cocoon moths. You can see the cocoon here. Here's what the mine looks like. It's a tiny little mine. They get out and feed on the surface a little bit after that. This turns out this is a new species. It's turned out to be a new species. We're going to describe that new species hopefully before too long. We'll see how that goes. And then another one that could be a new species is a stem miner. You can see these red trails. That's made by a moth, a kind of marmora. We've got one adult or a few adults from that. We'll hopefully figure out what that one is before too long. And this is all in North Carolina. Yeah, these are all in North Carolina. Yep. Now, I don't know about throughout North Carolina, but definitely it's in the Piedmont quite strongly, and on the coastal plain, at least some of the coastal plain. All right, so here's so a lot of these leaf miners are overlooked. I want to bring an example of a plant called Symphorocarpus orbiculatus, or coralberry. There are many websites out there showing plants, and on these plants are pictures of leaf mines as well. So the damage actually stays long after the miner is gone sometimes, and so you can see them. And a lot of people sort of don't think about that, meaning that there are maybe natural history websites on these plants, but they don't mention, and they might mention things that eat the plants, but they don't mention the leaf miners because they just don't see them. So they're too small for people to notice a lot of times. So these are two separate websites, same plant species, and actually they are all have leaf mines. And so some of these, there are multiple leaf miner species on this plant, and I'm working out what some of them are right now. All right, so how I became addicted to these things. Well, there was one that I found on an evergreen plant. I wanted to use it for a lab for my classes, my ecology class. And so I tried to identify it, and that was the first time I tried to identify a leaf miner. I sent samples to the NC plant disease clinic, and they didn't know what it was. And a friend of mine introduced me to Charlie Eisman. Charlie Eisman's working on a book on these things. You should look him up. If you are interested in these things, you should look him up. He does a lot of things with small critters and has written a book on tracks and signs of invertebrates. He's an amazing guy. And he looked at the photos and said, it's probably a new species. And my jaw dropped because I thought, wow, I never thought I would discover a new species in my lifetime. And here's a picture of the larva inside it. And they mine into the stem and down. So I was really surprised. So I thought, well, I'm going to try to look for as many of these as I can. And I always thought that if you wanted to look for new species, you'd have to go into the deepest tropical forests or the deepest oceans. But maybe all you have to do is go into the deepest roadsides or the deepest backyards. And in fact, I've found and helped document new species in both of those kinds of places. One of the things that I will take a short break and say is that this roadside has recently been sprayed. You can see all the plants are brown. If you spray the plants with herbicides, then all the insect associates die as well. They have nothing to eat. And so one of the things that I'm worried about is the Department of Transportation and Duke Energy, Progress Energy, whatever, are spraying the roadsides and under power lines rather than mowing. And that may save them money, but it is destroying our biodiversity. in the state, and I am kind of worried about that. At least there's some suggestion that it probably does, and given that if they're killing the plants, they're going to kill the insects that are associated with those. And then deer are another threat to biodiversity, potentially too. There are more of them than there used to be, and they're eating some plants over others, and some plants don't really have a hope of getting by with deer will eat them very quickly. So I'm going to talk about some of the discoveries that I've made with regard to leaf miners. And this is in conjunction with Charlie Eisman and other people. This is a collaborative effort. The range expansion, so new locations for miners that are well known, or miners that are well known, but it's a new host plant, a new food plant that we didn't know they ate, or an entirely new species. And so if we're looking for these, which you're going to be looking for these as well, where do you look? Well, look everywhere, but look closely. Pick unusual plants, for instance. If you find a plant that you don't normally see, look at it and see what's eating it. Maybe it's going to be something new or interesting. I've found actually new species on common plants. Plants like Baccharis, saltbush, plants like winged elm. Some of these common plants have yielded new species of leaf miners. So really just look anywhere and everywhere. Yes, yes. So here's a range expansion, an example of a range expansion. So Gelsemium sempervirens is a common plant around here, Carolina jessamine. And here's some miners I found on it. You can see the mines, they're just under the surface. This is more of a sap feeder, so it has really long mines, but it actually ends up being a very tiny miner. They're just eating a lot of sap, and it takes them a while to grow. And then here's what the adult looks like. It turns out this hadn't been found north of Florida before we found it in North Carolina and South Carolina. So, and not all the range expansions I've found are northward expansions. Some of them are southward expansions. So they may have been here all along, but we just didn't know that they were here. So there were some that I found that hadn't been found south of Kentucky before, things like that. So there are many different, and this is a fly. So there are a lot of the ones I'll talk about initially are flies, but there are moths as well. We haven't found, there haven't been many sawflies that have turned out to be new, just a few sawflies. Most of the diversity of leaf miners are flies and moths. So I'll start with flies and then I'll talk a little bit about moths. All right, so here is a lawn. This is a lawn. Some people would say it's not well taken care of. I love it because there's more biodiversity. If you were to magnify this plant right here, oh yeah, they do harm the host plant, they're parasites, but usually plants can, there's only very little indication that very little data showing that in very few cases they're known to hurt the plants. In other cases, it's probably minuscule damage, like they probably don't, there's probably not easy to measure the damage that they do. It's a small amount of damage, but still. So we can magnify these plants. This is corn speedwell. And if you magnify this leaf, there's a leaf mine on it. It's a tiny little leaf mine belonging to a fly called Phytomyza crassiceta. And it turns out this is a new host plant for them. Corn speedwell wasn't a known host plant for them before. It turns out there are 3 species in the same genus, one of which is native, 2 of which are not, that they eat. And so that people didn't know they ate before. There are other ones that people knew they ate. So this isn't the first record, but I found some first new host records for some miners, but not this one. All right, so here's another host range, some host range documentation as well. This is on a plant, was found first on a plant called Carpephrus boedifolius. It's a beautiful plant in the sandhills and coastal plain. And the mine extends from the top of the leaf to the bottom of the leaf. You could see it's sort of, this is just one surface of the leaf. It's just, if you turn it over, there's more of the mine. And it turns out to be on other plants as well. You can see the larva here, this little yellow thing is actually the larva. And so it eats other plants as well. We're still figuring out all the plant species that it eats. It may eat a bunch of different things, but because it was discovered first, I discovered it first on Carpephrus, we called it Liriomyza carpephryi, named for the plant that it eats. And so here's what the adult looks like. Again, it's a fly. You can tell it's a fly because it's got two wings, one pair of wings, and then this thing right here that looks like a little club is called a haltere. It helps it stabilize it in flight. All right, so this turned out to be a new species and as well. And so now when we're talking about new species, how do we get to name new species? Well, there are three considerations. The first one is the species really new? Can we convince ourselves that it's new? And then how do we convince other people that it's new? In other words, we have to actually formally describe the species using the characteristics that are different from known species and publish it so that other people can know about this species. And we also have to choose a name for it. And how do we do that? So I want to talk a little bit about that process. And this is something that I haven't talked about much before in public, so let me know how it goes. I talked with Owen Lonsdale, who's helped us describe the fly species that we found. He's in Canada, and he's really cool, and he talked with me about a few challenges to knowing that the species is really new. One of the challenges is how well studied is the group? If the group is not well studied, then it's really hard to figure out how many species are in it, and which species they are and how to distinguish them. So you have to study them for a while before you can figure it out. The second thing is how diverse is this group? The group of flies that makes up most of the leaf mining flies in the US is actually about over 800 species in the US alone. And so that means that it's really diverse. And so that makes it challenging to figure out, is this species really different from all the other ones that are out there? And then we have to find characteristics that tend to vary by species in that family, and they may be different from family to family. So they could be external characteristics, but it might not be something as easy as color. Color might not tell you very much, although sometimes it does. Or a combination of things. Usually it's a combination of many, many characteristics. And male genitalia is one of the characteristics that tends to vary a lot, because a lot of these organisms have lock and key mechanisms where the genitals fit together in very specific ways. And so if you have ones with different genital characteristics, then it's possibly different than something else with different genital characteristics. And we'll talk about, someone asked a question, we'll talk about that in a minute. We can build family trees of these organisms. Here's a bunch of organisms in the genus Liriomyza and how they're related to each other. And we can learn things from these family trees using all the characteristics we've determined. And one of the things we might learn is this red species, Liriomyza cicena, is actually potentially two species, because we've got one cluster over here and one cluster over here. It may be at least two species when we thought it was one. So maybe we should be looking harder and we look at those characteristics. The characteristics actually separate those species out. All right, so here are two species of flies. You can see some differences, duller versus more shiny, whatever. There's a few other characteristics. These are both in the genus Cerodantha. But when we look at the genitals, we can see even more differences. This is the external genitalia parts. Then there are these are the internal genitalia parts, including the phallus here. And so side view, top view. And so the shape of these things can tell you a good bit about how different they might be. And then looking at enough specimens to actually figure that out. So these were determined to be two different species. Again, species designations are to some degree a work in progress. If new data comes out determining that, well, we were wrong, there actually is a continuous variation between two species, we might end up lumping species. I don't know how many species there are in North Carolina, don't know, but there are hundreds in North Carolina. I've found at least 200 in one site, and then I found at least 100 species, more than 100 species in a couple of other sites, and there's not necessarily there's not complete overlap between those. So there are many, many species out there. All right, so if you have only 5 species in a group, it's pretty easy to tell them apart. In this case, each blob is a species, and there are characteristics that help us separate those species, so they're distinct from each other. But what if we discover a whole bunch more species in that same group. And yes, DNA can be used as well. Those are good characters. So yes, DNA can be used. It's not always preferable to external characteristics, but it is one of many tools. And so it should be used definitely in conjunction with other tools. All right, so when we discover a whole bunch more species, there's overlap between species. And now we can't determine whether they're different from each other using the characteristics we used to use. So we have to look deeper and look at more characteristics. And when we do that, we find that we can distinguish some species from each other. And so all these are now what we call separate species. We could designate them as separate species, but we thought this red one was different from this yellow one, but it turns out the more characteristics we look at, the more we just find overlap. And in fact, we can label this as one species. So looking in more detail can cause us to lump species as well as to separate species from each other. And natural history, this is to answer a question that came in before, natural history does matter. In fact, some of we can use natural history data like the host plant they use, when they come out, the seasonality, their mine characteristics, all of that adds more data. Sometimes it's not useful data, like sometimes all the mines look exactly the same and they're indistinguishable, but sometimes they are distinguishable, and sometimes you can actually use that to determine what species we've got. And so it feeds into it's just more data about these characteristics. One thing that's exciting about this work is that mostly when people would look at leaf mining insects in the past, they would use adults. So they'd collect adults and describe them from the adults. But that divorces it from what the actual critter is doing and what its habitat is. And so a lot of host plants were unknown. And so the exciting thing about this work is we're going out and finding them while they're larvae still in the host plants. And so we can tell what host plants they're in, what season they come out, look at their mine characteristics, as well as when we get adults, we can look at the adults and that come out. All right, and we can use all of that information to help describe the species. Once we have determined that they're actually new species, then we can list those characteristics in a publication and convince other people that they're new. So here's an example of a couple new species. So this is a species that mines on the Japanese wisteria. It's an invasive species, but it also mines on the native black locust trees. And it has a blotch-like mine at the end of the leaf. And what's cool about this one, this is kind of crazy. So this one, the larvae are active for about a week or so, and then they drop out and pupate in the leaf litter. When they pupate, they stay as a pupa until the next year and then come out as an adult and start the life cycle over again. So you have to wait an entire year. When we found these things, we had to wait an entire year to get adults. And here's what the adults look like. So this one's named for characteristics of its genitalia, its phallus. A stout phallus, it means stout. Anyway, so it turns out that when we looked at the natural history, when we looked at the mine characteristics, there was another mine of a different characteristic on the same types of leaves. It started as a trail and then widened to a blotch later down the leaf. Well, it turns out that we suspect that this is a new species, a separate new species, and we have yet to get adults that are in good condition from this one, so we haven't described it yet. But I collected some this year and they've pupated, so with any luck, we'll have adults next year. We'll see, and we'll be able to describe that species. This other one was described recently, within the past couple of years. So how do we name a species once we are pretty sure that it's new? Well, we give it a two-part name based on Linnaeus's binomial nomenclature. We fit it into the genus that it's part of, and occasionally if we find ones that are in a new genus, we have to name the genus, but usually we don't. Usually we fit it into the genus that it's part of, and then we give it a specific epithet. So it's a two-part name, like Agromyza. So I'll give some examples in a second. We can name them for external characteristics of the organisms themselves. We can name them for the host plant that they use. We can name them for the location where they were found, or people we want to honor. So all of these things could be problematic and require name changes later. Like for instance, what if the host plant's name changes? Well, should the insect's name change too? What if the location, what if we find a whole bunch more than we ever knew existed in a new place and we've named it for the old place where it's rare? Maybe we change the name or maybe not. But either way, some like Philadelphia vireos, it's a bird that's called a Philadelphia vireo. It only passes by Philadelphia in migration, so it's rarely ever in Philadelphia at all. Anyway, all right, so we can name them after all these things. We have to follow Latin or Greek rules depending on the etymology of the name, the origin of the name, or depending on the ending of the name that we're looking at. All right, so here's an example, Agromyza soca. Soca is Greek for stout or strong, as named for the phallus characteristics. Here are a bunch of different new fly species that we've described that are named for the host plants that they eat. This one feeds on Arundinaria, so you can see that in the name. This one feeds on Chrysopsis, you can see that in the name. And why all these different endings? Well, some of the endings matter in that some of them, like this one, Vora, says that it eats that thing, it eats the plant, it eats Polygala. These say that it's associated with Arundinaria. This is a little associate with on Arundinaria. Iella means little associate. And one of the reasons that names like this are chosen, why we might choose one and not another, is because one's already taken. So we look at which ones are taken, which ones have been used before, and which ones are open, and we decide based on all those things. And based on the ending of the plant name, we might choose one ending of our species name versus another one. All right, so here's one named for a place. This is named for St. Andrews University, where I work. I pushed for that one just because I wanted something named after our place, the place where I work. This one was discovered there. And Charlie, I had nothing to do with this. Charlie named a fly after me, Cerodonta feldmani, so I've got a fly namesake out there, which is kind of cool. I'm happy about that. It's tiny, you would never notice it, but anyway, it's cool. So I'll talk about some of the new species we've discovered. This is on wild bamboo or switch cane. I don't really like that name. I like wild bamboo better, but these are what the mines look like. And in those mines, we found several species. So they sort of mine together into one mine. They combine their mines into one thing. And we found 2 in one mine and 1 in another. And so we found at least 3 new species on that one plant species, which is pretty cool. And that was in southern North Carolina, so southern pines and or Aberdeen and Laurinburg. This one was found in Duke Forest on Polygala verticillata, and I've only ever found this one in that one site, but I haven't found Polygala verticillata very many places. It's not a plant you see that often, so if you know of other populations of it, look for this minor. It's pretty beautiful. And this is a minor that I've only ever found at Lee Farm Park. And so that's the thing. Most people aren't looking for these things. If people look for these minors and more people are out looking, the chances are we'll find more, hopefully. Or maybe not. Maybe we'll find that certain minors are only found in one place. But either way, it'd be nice to know that. Now, those are all flies. Now I'll switch to some of the moths that we've discovered. This one was discovered in a few different places, but it was first reared meaning raised to adulthood, from a material from Laurinburg area. And it's Phylognistus, so what it does is that genus, what they do is they mine on the surface, upper surface, or the lower surface of the leaf, just below the surface, and then at the end of the mine they fold over the leaf a little bit and pupate inside, and so they keep their pupae hidden rather than dropping out and going into the leaf litter like some of the flies that I talked about. These just pupate right there, and so it's hidden from most things, although if you know what to look for, you can go and look for them and actually find them more easily that way, so humans can find them more easily. This one, what this one does is it mines for a little while and then gets out of the mine and makes leaf rolls and feeds from inside the leaf rolls. And this is on styrax, which is another plant that you don't see that often, but when it's there, it's really cool. It's a beautiful plant, and it's a really beautiful miner too. All right, and this is one that I still haven't figured out how to get an adult from this one, and I may never figure it out. What this one does is it goes through the leaf and then down into the stem, and I don't know how far down it goes or when it comes out or where it's going to come out. And I've tried many, many different ways of trying to get an adult, and I've never succeeded. So we'll see if I ever do. But I have succeeded on some stem miners. So this is one that mines down the trunk of a tree. There are several miners that do that, and I was lucky enough to find an exit hole, and when I looked around the trunk, lo and behold, I was totally lucky. There are some cocoons, and here's what a cocoon looks like. It's amazing. What they do is they spin some silk and then kick out these things called frothy bubbles. I don't know what they are or what they do, and outside the silk, and then they pupate inside. Maybe it protects them, but again, we don't know how. There are a few genera within this group of leaf mining moths that do this kind of thing, but don't know why. Here's what the adult looks like. So it's kind of cool. A very similar adult was gotten from sweet gum. We cut stems and I put them in water and in a bag, and it exited and came out and pupated in the bag, and we got an adult from that. Maybe the same thing as the other one, we don't know yet. They look very similar, so I don't know, but what the DNA will tell us or the genitalia will tell us. I look for them everywhere I go. When I took a student down to South Florida for a conference, he studied spiders with me, and we were so excited to find hedges in this hotel. It was beautiful. The hedges have native species in them, and the native species have leaf miners on them. And this one belongs to a group that's around here too, but the species is different, but the group is the same. What they do is they mine, and then they cut a hole around themselves and drop in that leaf package. They drop into the ground, into the leaf litter where no one will ever find them. And they spin a cocoon inside that leaf package. It's really cool. So this turned out possible new species or new country record, not sure, but we'll see. It's on buttonwood. So there are some really new discoveries. I've been looking at this plant, Gelsemium sempervirens, for years and years now, and I've never found a stem miner on it. And then just this past fall, I found a stem miner. Here's the caterpillar. And I have a movie of it feeding, I'm not showing it here, but still. Either way, it's pretty cool. We got some adults from it. I don't know what they look like yet, but I'll hopefully get a photograph of them soon. Either way, it's pretty cool. A new species, probable new species, or we'll see. At least it's something different than I've ever seen before. I've looked at wild ginger plants for years and years and looked for miners on them for years and years. And then just a week and a half ago, I found a miner on Hexastylus. I've never seen one before or since, and we'll see if I ever see them again. But what it does is it gets out of the mine and feeds on the surface. So we'll hopefully get an adult from this. This is totally new information. I don't think there's any record of anything that eats Hexastylus out there, but we'll see. Sometimes the adults are already identified, and sometimes they're not. Sometimes they're not. So, there are lots of different things out there that people luck out and get or don't get. So if you want to look for these things, you have to use magnification. That's very helpful. A dissecting microscope is fun, but not necessary. So just any magnifier will do, even binoculars turned backwards. Look at different times of the year. Check out different plant species, different plant parts, stems and leaves. And if mines are on leaves, you can photograph both sides. That's helpful, whether it's on the top surface or the bottom surface, that helps. Or backlit leaves, you can sometimes see larvae inside. You can collect the leaf and put it in a vial with a little bit of paper towel, with a little bit of water to keep moisture in. You label it, you can sometimes, rarely, some species need to pupate or bury in soil to pupate. That doesn't happen very often, but if it's wandering around a lot, you might try. And so I can give them a little bit of soil to pupate in. There's a paintbrush for moving larvae around. For preserving specimens, you can use ethanol for most groups, but for moths, their scales fall off, so you need to use empty pill capsules to hold them. They're tiny in there. Or coin envelopes for pressed leaves, for mined leaves. And so those things can help preserve specimens. You can submit them to iNaturalist. So you guys are going to use iNaturalist if you do the assignment afterwards. All you need is a photograph of the organism, a location, and a date, and you've got stuff that scientists can use, information that scientists can use if it's identifiable. I submit to Bug Guide because that's what my colleague Charlie uses. So I submit to bugguide.net. Accounts on each of these things are free. You can submit the photographs and you can get updates on what they turn out to be, if people can identify them. Yeah. And so one other thing that we should be, if we're wanting to do this stuff, is to advocate. We have to advocate where we can to preserve biodiversity. So I'll bring up the roadside spraying as well. In that case. I see some other questions. I'll address those in a second if I have time, but I'd like to acknowledge a lot of people who helped me find species, who helped me identify species, who helped protect areas, and who helped with access to field sites. And if there's any questions, there are some, I'll take them if there's time. So let's see. Yeah, there are lots of examples of larvae waiting to be identified. There are several, or adults that have come out that we haven't identified yet. There's a backlog of moths because moths, there's a lot of work still left to be done identifying or revising groups where there are a ton of new species coming in, and people have to do what they call revisions of the group to actually revise the groups and figure out who's related to whom and what they are. So there are a lot of different examples of those. Yeah, there are many. I'm not sure how many right now, but I could give you that if not. So other ecological connections. Oh yeah. So right. So I don't know about diseases, but there are examples of leaf miners that team up with bacteria that to help leaves stay greener longer, or microbes to help leaves stay greener longer. So if you see leaf mines and on a leaf in the fall or a leaf that's fallen from the tree, and everywhere on that leaf is yellow, but the area where the mine is, that's possible that they're teaming up with bacteria to actually keep the leaf greener, which is kind of cool. There's one documented case of that I know of, at least one paper where they discuss that. There are also, sometimes I get parasites of these things. Sometimes that they're identifiable, sometimes people don't, there aren't people out there to identify them, so sometimes we just release them, the parasites, in the place where we found them. Yeah, let's see. So diseases they pass, we don't know, but it's possible that they pass diseases to plants, maybe not to other things, but to plants anyway. Possible that they do, like some other herbivores do. It's possible they do. I don't know. We don't know anything about that yet, really. I mean, I don't know very much about it. There may be a few things out there, but I haven't encountered very much. We have, yes, I have videos, but I don't have them right here. If there's time, I can dig some up. What is a leaf miner on Eastern Columbine? There are two phytomyzas. They're both flies on Eastern Columbine that I've found in my yard. And I don't remember their names right off the bat. They're both named for the host plant. So Aquilegia vora is one of them, and Aquilegia foliella, I think, is another one. I forget, but there are two. The one that looks like more of a trail, and one that looks like a little more of a blotch. Either way, I think there are two. Yeah, there are some cases where there's measured harm to the host plant, but I don't know of any cases where they've been documented to kill host plants. Usually they're feeding on just such a small portion of the leaf, but sometimes they're on every leaf. Sometimes there are multiple ones on a given leaf. So they could do damage to these plants, but I don't know of many cases where they've actually been documented, the damage. All right, so I've got about 1 minute more. I guess, let's see. So are there particular host plants, local areas that you're particularly interested in? Every single one. I mean, basically, I started doing this and trying to help Charlie Eisman out, and it's been really fruitful. I feel like I'm getting a handle on the sites where I go at this point. So like Lake Crabtree County Park, I spend a lot of time there. I spend a lot of time in Laurinburg on the campus where I work, and Lee Farm Park. But, you know, there's a lot of other places out there, and I have no idea. You know, there are sometimes I find a leaf miner in one place, in one location, and not in others. So if you're looking in a different location than I've been, chances are that you'll find things that people haven't found before. That's possible. So even if you haven't found, even if you don't, you'll still potentially expand our knowledge of it and the range of it and how many sites it occurs. So I think all of it's valuable knowledge. And so the assignment that I give is geared toward, if you want to do it, is geared toward finding some of these things and reporting on them on iNaturalist. If you want to submit them to Bug Guide as well, that can be good too. The websites where you submit them are actually in this document, in the slides, and I assume those will be available. Also, the recording of this will be available. So I can also send some video files of larvae feeding to Lauren, and she can post those if you want to. You can post those. Yeah. All right. So yeah, please feel free. There are lots of lessons you can do with this. If you want to brainstorm lessons to do with middle school students or other people, other groups of students, let me know. I can help. Yeah, you can help brainstorm ideas if you want.
Well, and Tracy, I just have to say thank you. I'm going to pop in here and say you have personally blown my mind about the creatures that might be living in my yard that I had no idea. If you are in the same boat, for all of you here on the webinar going, I came because, you know, maybe I was interested in EE credit for the North Carolina Environmental Educator Certificate through the Department of Environmental Quality. If you're here for that and staying to the end and going, how do I get my credit? I'll send this to you in an email. But if you just enjoyed this webinar and had a great time learning about leaf miners, it qualifies for 1 hour of criteria 3 or continuing education. And I'll send out all the directions for how to get that, just if you want to watch the webinar. But basically you fill out the EE form and just submit it in. And I'll have a link to that. But if you're here going, I wanted to get the criteria 2 credit, well, this is where it's your opportunity to go out and do some observation. So as Tracy has mentioned, we do have a homework assignment for you where you're going to spend some time outside using iNaturalist to try to find 10 herbivores or 10 signs of past herbivory.
Actually, I think we read it to 5 or something.
Oh,
is that right? But if you want to do 10, please do 10. Like, do as many as you want.
Yeah. Yeah. So, okay. Because the one I have says 10 or an hour, whichever comes first.
Yeah, that's right. That's right.
Many people, everybody.
Yeah, maybe we did that. We just did time. That's right.
Yeah, everybody has very different access to natural spaces, and who knows what kind of leaf miners you find. So spend some time out looking.
I found them on plants in sidewalk cracks in cities.
Awesome. So they're going to be around. So check it out. Look around. If you enjoyed this webinar, this is part of a series that EENC is doing. We're doing these on rotating days of the week at 1 o'clock. So next week, I believe is on communication. We've got one after that on group management. So we hope to see you again in the future. But other than that, thank you for spending an hour and we'll be in touch.
Thanks.
Thank you.
Good stuff.
Thank you.